So, today's lesson is about the measurements of the diameter of animal cells of plants by use of eyepiece graticule and mic. Here in the diagram, there are two scales. The upper scale is micrometer, in which each division is 0.1 mm, 0.1 mm. Mostly 0.1 mm scale is used, or it can be 0.01. So we have to look at in the exam questions whether it is in 0.1 mm or 0.01 mm. So I have chosen 0.1 mm here. So one length starting from one division to the other division, so from here to here, it is 0.1, 0.1 mm, and this is a micrometer. A micrometer term is used in biology and in physics as well. In physics, the security gauge micrometer we are using here. So here is micrometer or 0.1, right? So the second scale is the eyepiece graticule here. This one is the eyepiece graticule. And this eyepiece graticule has divisions, 100 divisions here. Eyepiece graticule. Correct. Has 100 divisions without any kind of requirements. So there is no unit of this divisions. So we have to calibrate to find out the length of one eyepiece graticule. So how can we find one division of eyepiece graticule? So the method is that we have to superimpose these two scales with that of microscope and that calibrate it. The first step of calibration is that we have to uh, match the dimensions of the micrometer with the eyepiece graticule, and we have to look at that how many divisions of eyepiece graticule within this 0.1 mm. So it is from 50 to 100. This we have to check. Or from 0 to 50, it is 0.1 mm here, 0.1 mm, and it is 0 to 50. So that is 50 degrees. So 50 to 100 also 50 degrees. So it means that we have to write here 0.1 mm equal 50 millions. EIP, billion ball, I get EBT. Again, we get in here. So if we look again here, from 0 to 10, then 20, 30, 40, and 50 in 0.1 division of the micrometer. So if we want to find out one division, then we have to divide 0.1 by 50. So here we can write 0.1 divided by 50 divisions. So it will be equal to, you can use get two quarter, 0.1 divided by 50. That is 0.1 mm. So what one eyepiece graticule, one eyepiece graticule or 1 deviation white is equal to 0.002 pattern. This is the you can say second step. First, we have superimpose these two scales, micrometer and the eyepiece graticule. Then we have to look at how many deviations of eyepiece graticule is matching with the 0.1 or smallest deviation of micrometer. That was 50 deviations. So we find 0.1 divided by 50, 0.002 and get one eyepiece graticule length. The third step is that we have to superimpose this eyepiece graticule on a slide of specimen cell and find out the deviations of the eyepiece graticule, that how many eyepiece graticule matching with the cell. For example, if we get draw a cell over here, suppose we have cell here and this cell is of this shape, and we can find here that it is starting from 21 and ends at 61. So it is the maximum width of this cell. We have to superimpose with the help of light microscope. So what are the total deviations here? It is starting from 21, 39, and then there is 19, 48, then up to 29, and 39, and then we can tell there is 40. Just one division about it is 61 bracket album. So plus minus 1 it can be error. So it is 41 altogether. You can 40 is it is altogether 40 deviations. 21 to 60, 40 deviations of the eye study. So what is the third step? That we have to multiply 0.002 mm to 40. Or we can convert this 0.002 into micrometer, then multiply. Or we can convert at that to 0.002 mm. If we want to convert it to micrometer, then we have to divide it by 1000. It will be 2 micrometer. So then we can multiply it. We can multiply it and multiply by 40. It will be 80 micron. So it is the maximum width of the cell study. 21 to 61. So this one is the 80 micrometer here. So the process is we have to convert this 0.02 into micron, which is 2 micrometer here. We have divided by 1000. After converting to micrometer, we know 1 deviation is 2 micro, then how many 40 deviations are there? Then we have to multiply it, that is 80. This is how we can find out the diameter of plaque cell, any plaque cell. Here is an example of measuring the diameter of different organelles or as a whole cell by the help of eyepiece graticule and micrometer. So in the question, it is mentioned that it is a stage micrometer here. So this one is a stage micrometer, and this one is the eyepiece graticule here. We can label it here that it is a stage micrometer, and this one is the eyepiece graticule. Is graticule. And the other thing is mentioned in the question that it is 0.1 mm stage micrometer. So it's 1 division is 0.1 micron, 0.1. 1 division mm, and it contains 10 to 50 here. So altogether there is 40 divisions of eyepiece graticule. So last time we have discussed that the diagram which is given to us is 0.1 micrometer containing 50 divisions, and we have calculated accordingly that one eyepiece graticule is 0.2 micrometer, 2 micrometer. So here we have to divide 0.1 by 40 to calculate one eyepiece graticule. So here is the calculations that we have 0.1 mm, that is equal to 40 divisions of eyepiece graticule. EPG, you can write EPG, eyepiece graticule. So we have to divide 0.1 divided by 40, and we can use calculator to measure this. So use calculator. 0.1 divided by 40, it will be 0.0025 mm. Previously we have done 0.1 divided by 50, but now we have divided by 40 because the diagram given in this question have 40 divisions in matching with 0.1 mm of the stage micrometer. So we have divided by 40, so it is 0.0025 mm. So next one is that we have to convert it into micrometer. So 0.0025, we have to multiply it with 1000. So it will be in our micrometer 2.5 micrometer. So one eyepiece graticule, one EPG division is equal to 2.5 micrometer. Now we are this is calibration process, and this calibration process, the value we will use to solve this question here. And in this question, you can see the stage micrometer is replaced by this plant cell. And this plant cell, you can see here is a cell wall here on the outer side here. And then inside you can see there are chloroplast. There is a large vacuole which pushes the chloroplast, and the nucleus is here, which is the largest one. And here is the chloroplast. So in the question, we have to look whether they are asking about the maximum width of the chloroplast or the nucleus. So both we will calculate today. So here they are mentioned here nucleus. So how can we calculate? First we have to find here the maximum width here. So by the help of lead pencil you can mark. Then with the help of scale you can draw lines so that we can find out how many eyepiece graticule divisions are there. So it is about 3 here, 5, 8, 9, 10. So it can be 9 or 10 divisions are there. So what we have to do, we have to multiply it now. It is the width of or diameter of nucleus. So from the calculations of the calibrations, we know that one eyepiece graticule is 2.5, and how this is one eyepiece graticule, one EPG. And now we have 10 EPG here, 10 EPG. So 10 EPG means 10 multiplied by 2.5 micrometer. It is equal to 25 micrometer. So the width of the nucleus is 25 micrometer. This we know that it is nucleus is always larger than 10 micrometer. So it is 25 micrometer here. So in the answer we have to look for the value closest to the 25 micrometer. So it can be written over there 20 or maybe it is written 26 like that. But not, it should be further away. The value should not be further away from this answer. 25. Same if we want to find out the maximum width of the chloroplast here. The chloroplast is in surrounding the, for example, here is one chloroplast we can mark here. So this one is one of the chloroplast. You can choose any. So this contain from here third divisions to there are 3 here, and here you can mark here. So 5 or 6 divisions are there all together. Is 5 to 6 mean that if you multiply it 2.5 with the so here is the diameter of chloroplast. So it is 5 divisions here, 5 divisions of eyepiece graticule, which we mark on the diagram, and multiply it by 1 eyepiece graticule division, that is 2.5 micrometer. So it is now 5 multiplied by 2. We have to use calculator again. 5 multiplied by 2.5. So it is 12.5. 12.5 micrometer. It's the width of or diameter of the chloroplast. One of the chloroplast. You can check. It can range from 10 to 14 different chloroplast here. And the value in the answer we have to look for, if there is MCQs, then it should be closest to this 12, maybe 14, maybe 10. So it can be right answer. But if it is further away, then you cannot choose that value. So this is how we can find the diameter. Even we can find the total width of this plant cell, which is in lengthwise. So here we can mark the cell wall here. This one and this one we can mark here and find out here approximately 1 plus minus divisions. So it is starting from 10 to 89 or 88, you can say. So it is 78 divisions are here. So 78 multiplied by 2.5, you can calculate 78 multiplied by 2.5. So it is 78 divisions, and 1 division is 2.5 micrometer. So 195 micrometer, it is total micrometer is the total length of this. From there you can find out magnifications, even if the values of actual width is given of this plant cell. So magnification m is equal to I over A, you can find m is equal to find out if some questions are there of magnification m equal to I over A. So A will be given actual diameter of this plant cell or actual diameter of the nucleus or whatever. So you have to calculate image size by this calibration process by use of eyepiece graticule and micrometer. Then you have to divide it by the actual size so you can get magnification easily. So this is how it's got. So if you have some questions, you can just put in the comments. We can answer that. See you next time. Thank you.